Hi, everybody. I hope you're enjoying your beautiful Sunday afternoon. This is the second of the two lectures um, from the immunity, inflammation and immunity section of the of pathophysiology class. Um, the lectures that you'll find down in the media library are older lectures, but I think they're still valid and still pretty good. So take a look at those for detail. I'm just going to go through this second PowerPoint, the one on altered immunity, just briefly to give you an idea of what to focus on, where to put your energy. There's a lot of information. Keep in mind that when we talk about immunity, that first lecture, inflammation, is the generalized approach. When something causes damage, when when there's an illness, when there's a environmental toxin, when there's a toxin that's produced within your body, when there's a virus or bacteria, all of those things lead to inflammation. And we discussed that in the last lecture. In this particular lecture, now we're going to be working much more specifically. So we're really targeting antigens, foreign bodies, foreign materials, especially foreign proteins that enter our body and cause problems. And so our body neutralizes those or figures out a way to deal with those. So that's what we're talking about here. So if you look at these first few slides, again, this is that third line of defense. Things are much more specific. There is, again, the recognition of antigenic material or foreign material, and then the quality of memory. Your body has the ability to memorize what that antigen was and to create a very immediate response on subsequent interaction with that antigen. So we talk about two parts of the adaptive immune response, the humoral mediated immunity and cell mediated. Humoral simply means that which is circulating in the fluids, and that refers to antibodies. Cell mediated means that cells are primed. Specifically, we're talking about T cells, lymphocytes that are activated and that have the ability either to call on other parts of the immune system to say, hey, there's something going on, there's a foreign invader, alert, alert, or directly to cause killing of that, either that bacteria or that cell that has been damaged. So that's what cell-mediated immunity is. Again, the cellular components, you should go through those and making sure that you understand what T cells are, what B cells are. NK cells, we talked about those in the last lecture. NK cells are non-specific and non-specific lymphocytes that attack cancer cells. They're natural killer cells that attack cancer cells or virally infected cells. So that's their whole point. They're not as specific. They just see that a cell has been infected or affected in some way. How does it know that? Well, it can read what's happening in the outside of the cell. When a cell is affected, it presents antigen on the outside, and your body recognizes that. Certainly NK cells do that. They recognize this isn't right. I can see what's going on in that house, and I don't like what's going on in that house. There's a flag in the window of that house that's telling me something is inside that's not right, and it will destroy that house. T cells and B cells do a similar kind of thing, depending on the type of T cell, and then B cells act slightly different. We'll talk about those momentarily. You can read about T cell maturation. T cells, as you know, and B cells, all lymphocytes are they're born, if you will. They develop in the bone marrow. B cells migrate to certain parts of the bone marrow for maturation. T cells migrate to the thymus gland. That's why they're T cells for maturation. And from there, they make their way to the secondary lymph organs where they hide out and wait for presentation of encounter with antigens. All right. So this slide, immune defense concept map, take a look at that. That sort of summarizes what I've just sort of described earlier. Look at the difference between active immunity and passive immunity. Active is what your body does. Passive is when you directly give antibody or directly give immunoglobulins or T lymphocytes. You can directly give T lymphocytes that have been sensitized or to a substance previously and that can cause a known reaction. So make sure that you just look at that one for definition. Again, looking down there, lymphatics, that's helpful to look at, but just don't spend a lot of time there. Adaptive immunity, talked about that slide there, cell-mediated immunity versus humoral immunity. Now we're starting to get a little bit more specific. So when we talk about cell-mediated, again, we're talking about lymphocytes, T lymphocytes, and there are two different types. The cytotoxic lymphocytes, which as I alluded to earlier, cytotoxic T cells cause a direct killing of the affected cell, whether that is one's own body cells that have been infected by a virus or have been manipulated in some way, or are cancerous cells. They're not playing by the rules any longer. They look weird, and your body says, yeah, this isn't right, and causes that direct killing. The helper T cells are the other type of lymphocytes, and those are, they amplify the immune system. They call on other parts of the immune system. Either they tell the T lymphocytes to, hey, we've got a problem. I need to tell you a little bit about this, and they inform the cytotoxic T cells, and they help support the cytotoxic T cells to do that killing, or they cause maturation. They help the maturation process of the B lymphocytes. The whole goal of B lymphocytes, humoral immunity, is to mature into plasma cells. B cells become plasma cells. B cells become plasma cells. Make sure you understand that and know that. Plasma cells create antibodies. So B cells create or mature into plasma cells, which create antibodies. T helper cells, and specifically T helper 2 cells, T helper 1s are the ones that help out the cytotoxic T cells, the effector T cells. T helper 2 cells activate B cells to become plasma cells to create antibody. All right, so make sure that you understand that, and then we'll talk about more in detail. So let's see. Okay, so helper T cells. Helper T cells are again they're T lymphocytes, and they magnify, as it says right there, magnifies the interaction between the antigen presenting cell. So when you have antigen that enters the body, maybe it's a virus, maybe it's a bacteria, maybe it's a toxin that a bacteria has produced. It's some sort of foreign substance. Sometimes these are too big and they have to be broken down. You got a Pac-Man, phagocytize that substance, break it down, and then you take a little piece of it and you put it on the outside in the cell and say, it says, look at what I have in here. Look at what I've got. Okay? That's a signal, a little flag on the outside of the house, if you will, that says, hey, come on over here. Look what I've got over here. I've got a foreign invader. So that's what they do. There are some times when an antigen is too small. And so it's actually too small, and so it actually needs to be have another piece added onto it, and we'll talk about that's a hapten. Might want to take a look at what haptens are. Anyway, so helper T cells, getting back to helper T cells, so on the outside of their cells, they have what's known as a CD4 marker. It's just it's a protein, but it designates that as a helper T cell. Cytotoxic T cells have CD8 markers. So sometimes we refer to helper T cells as CD4 cells, and we refer to effector T cells or cytotoxic T cells as CD8 cells. And so sometimes we look at the ratio of CD4s to CD8s to see what's happening. Anyway, CD4 cells. So CD4 cells, CD4 helper cells can be recognized by something called a major histocompatibility complex, an MHC protein. And so I want to talk about MHC. So MHC, there's two different classes. You might have to skip to a couple of slides here. But the major histocompatibility, those are glycoproteins on the outside of a cell. And all cells have them except for red blood cells. Red blood cells don't have MHC proteins on them. There are two major classes. There's an MHC class 1 and MHC class 2. There was also an MHC class 3, which are actually complement proteins. Whole different function. Don't worry about that at this time. Anyway, these MHC proteins are encoded by genes on chromosome 6. And they're specific. The MHC class 1 and MHC class 2 are on different cells. MHC class 1 are found on all nucleated cells except for red blood cells. So they're mostly involved with virally infected cells and or cancer cells. So MHC class 1, interacts with the cytotoxic T cells, the CD8s. The CD8 marker protein can interact with MHC, with cells, antigen-presenting cells, that have MHC class 1 on the outside. And they've bound, they Pac-Man, they phagocytize an infected cell, a cancer cell, they digest it, and they put something on the outside that says, look what I have in here. And along comes a cytotoxic T cell, a CD8, which can bind with the MHC class 1 protein and cause destruction of that cell. Yay, that's what we wanted to happen. Take a breath here. MHC class 2. MHC class 2 proteins are recognized by, they're found on antigen-presenting cells such as macrophages, dendrocytes, and B cells. Mostly, you want to focus there on the macrophages because they're the ones that are doing most of the Pac-Man phagocytizing, where they engulf certain materials, they digest them, and then they're going to present. And they're a little bit less specific. They are non-specific about which antigen they choose. However, so they phagocytize, the macrophages phagocytize and engulf a antigenic substance, digest it, take a piece of it and put it on the outside of the cell, and that is able to interact with, I should say it this way, your helper T cells, which have a CD4 marker on them, protein on them, are able to interact with the MHC class 2 protein. So the CD4 interacts with with the MHC class 2 protein found on the macrophage, and that signals, that causes a signal of amplifying the immune system, saying, wow, hey, there's a cell over here, that's got something in it, and we need to create a response to this. Okay, so it calls on other aspects of the immune system to say, hey, we need help, we need help, we need help. Okay, so that's what's happening there. Now you have a couple of slides, maybe, I think you do, that show antigen presentation, actually what's happening and the interaction between the CD4 and the MHC2 and the CD8 and the MHC class 1. If not, go to YouTube. There's all kinds of videos, animations showing antigen presentation. Take a look at them. I think they're worth looking at so that you really get it in your mind. What's really interesting, well, maybe I'll come back to that. Sorry, I have a slide that will come back to that. All right, so then let's talk about antibodies. Again, I'm just skipping over a few things here. You can certainly read for more detail or listen for more detail on the lectures in the media library. There are a number of lectures there. So, endless amount of fun. You need to understand the different classes of antibodies: IgG, IgA, IgM, IgE, IgD. All right. And then antibody structure. So an antibody structure looks like a Y. You can see that in the illustration here in the PowerPoint, where the bottom part of the Y attaches to the cell and transmits information to the cell that gives about whatever it is about the foreign antigen that's attaching. So it looks like this. The antigen attaches up here, okay, and that transmits information down here through this the crystalline fragment down into the cell, which is down here, and then it tells the cell, it gives the cell information. So take a look at that. That's really important. You'll see that there is there are two chains to that Y. They go like this and then like this. The inner chains, because they're longer, are the heavy chains. The light chains are the two chains on the outside. All antibody structure have the same structure. All antibodies of the same class, I should say, have the same structure, except for the part up here. The part up here is what changes and binds specific antigen. Let's say it's antigen from goldenrod pollen, or pine pollen, or some kind of bacterial fragment that will bind. All of these are specific to that, but the rest of the antibody is similar, is the same. It doesn't change. So, okay, so then let's talk about specific immunoglobulins. I'm not going to spend a lot of time on this because you can read this, but making sure that you understand that the most abundant class, most antibody, is IgG. About 80-85%, as it says there. The other thing that's important is IgG crosses the placental barrier. So mom can confer that in any exposures that she has to the child. So the child has natural immunity. It has natural IgG early on for protection. All right. There are 4 classes. I don't care about that. That's too specific. IgA. So IgA, importance about that is it is a dimer. Instead of that Y, it is a Y connected to another Y. So there is a I can't do it. A Y connected to a Y at the base, and so it's called a dimer. So whereas IgG exists as the Y, IgA exists as the Ys, the bottoms of the Ys connected to each other. So they're found actually in circulation, oftentimes in secretory. There's something called secretory IgA, so they're found in the secretory juices, if you will. They line the gastrointestinal tract, they line the respiratory tract, etc. And so, and then they're protected by, there's this J piece that prevents them from being broken down, from being digested in the gastrointestinal system by the gastric juices, the hydrochloric acid, which would naturally denature a protein. But there's something that protects against that. All right. IgA 1s and 2s, you can take a look at that, but don't spend a lot of time on that. All right, IgM. IgM is really weird. IgM is huge because it is actually a pentamer, pentamer being 5. So there are 5 Y chains all connected, and so it's a very large immunoglobulin. The other thing that's important is it is a first responder. It's the first one that shows up in the primary response, that first response to an antigen. It is synthesized in fetal life. So the fetus will, on exposure, will synthesize some IgM. IgD, the only thing you need to know, there's not much of it in circulation. And what I hadn't mentioned earlier is that B cells also have receptors on them that can recognize antigen on a antigen presenting cell. Well, guess what that receptor is? It's actually an IgD. It's simply an antibody on the outside of a B cell. And it can recognize some specific antigen. All right, and then there's IgE. We'll talk a lot about IgE a little bit later on, but one of the things I want you to recognize or remember from this slide on IgE equals parasites and allergens. Now, where have you heard that before? Allergens in connection to parasites. If you don't remember that from the lecture, you should remember it from the notes. What cell was it? Bingo. Eosinophils. So now what's the connection to eosinophils and IgE? Puzzle for you. All right. So take a look there, and we'll talk about that later. Then you need to understand primary and secondary response. So you have this initial exposure. It takes a while to create an immune response. You have an exposure, your body doesn't recognize it, doesn't even know what to do with it, it has to process that, and then the B cells have to differentiate and develop. So B cell, the helper T cells will recognize it because it's been presented on an antigen presenting cell. The helper T cells, which are CD4s, which interact with MHC class II, will recognize it. They'll tell the B cells, hey, you need to mature, you need to grow up here, there's a war going to happen. And then the B cells will become plasma cells. Plasma cells will create antibody. All right, so that's what happens. After 5 to 7 days, the antibody that's created, the first responder is IgM. Yes, you are right. Which is kind of weird because it's the biggest one. You'd think that it would be something more simply, but it's not. IgM is the first responder, IgG, then after a couple of days, a slightly slower response, IgG is produced in on about the same amount as the IgM. Follows immune response, and there's a graph, and I would definitely take a look at that. All right, then there's some slides on antigen presentation. I think those are, again, take a look at those. And I think, I don't know if my links are still good, but you can just certainly Google antigen presentation, MHC class 1, MHC class 2, etc. The one slide here on antigen processing and presentation, I think that the interesting thing about that, please take a look at that so that you understand what's happening. But when a CD4 helper T cell interacts with an antigen-presenting cell that has an MHC class 2 molecule on it, it activates. And in doing so, that cell will produce specific interleukins. Remember when we talked about interleukins? Specifically, you'll see that if it produces IL-4, that will become a helper, T helper 2. If it produces IL-1 or interferon gamma and or both, that will become a T helper 1 cell. So there's a differentiation between that. And how does that happen? Based on the cytokines that are produced. Remember, we talked about cytokines. Cytokines are just a communication. Well, what's really interesting is when IL-4 is produced, that will actually inhibit the production of T helper 1s, and the vice versa is true. So take a look at that, and you can see that. So your body has several mechanisms by which it induces the development of T helper 1s or T helper 2s, depending on what's needed. Cell-mediated response, causing the effector T cells to be activated so that there's direct cell killing, or humoral immune stimulation, where you're causing those B cells to be stimulated into the development of plasma cells to produce antibody, and that's the kind of reaction you're going to get. So that's a really important slide, and understanding that is really important. Then a little bit later on, okay, so there's some other interesting slides. I think you should definitely take a look at that. But then you'll see that there's this connection of IgE to the eosinophils. And how does that happen? So make sure you look at those because that's really important, is to make that connection. We alluded to it earlier. But what happens is IgE, when it's activated, will bind onto mast cells and eosinophils and cause degranulation. So in an allergic response, you're going to get flood of IgE. The same thing happens in a parasitic infection. So parasites and allergic reactions are going to cause, are going to bind onto eosinophils and cause degranulation. So that's the connection there. Make sure that you take a look at that. The next slide is on how about antibodies crossing the placental barrier. That you're talking about how IgG crosses that. So a newborn has IgG levels near adult levels. And I think that next slide is really important clinically, and that you'll see there about up until about 6 months, babies generally don't get a lot of viral infections or bacterial infections. If they do, that's pretty serious because they have immunity. But along there about 6 months, you'll see there's a drop in the baby's immunoglobulins that it got from mom. but it hasn't been able to, you'll see that dip there, it hasn't been able to generate its own immunoglobulin yet. And so that's the period of time you start getting those ear infections, you start getting susceptibility to little colds and flus, etc. So take a look at that. Oh, and also remember, Khan Academy is a really good resource for any of the immune system stuff as well. All right. The next thing that's really important is the hypersensitivity reactions. You've got to know those. The type 1, type 2, type 3, type 4, what are they? I go through those fairly specifically in those ancient videos, but they're not that ancient, they're maybe 5 years old. But anyway, take a look at those. Those are very important. Type 1 hypersensitivity reaction, IgE mediated. So type 1 hypersensitivity reactions, that's your common allergic reaction, where you get the release, the IgE binds to mast cells and causes histamine release, and then histamine causes all those symptoms of runny nose, watery eyes, sometimes bronchoconstriction, etc. And the manifestations are right over here. Itching, urticaria, that's hives, conjunctivitis, your eyes get red, all that kind of thing. Okay. And then we can do, there are some tests that we can do, and you might want to just take a look at some of those various tests that we do for type 1 hypersensitivity reaction to see what is this person allergic to. I'll let you look at that. Make sure that you look at that that little slide that shows sort of the illustration of a type 1 hypersensitivity reaction and the IgE antibody. Type 2 hypersensitivity reaction. Type 2 is cell specific, in which there is a cytotoxic, there's a direct killing of the cells. This can it be immediate, 15 to 30 minutes, or it can be a slow reaction. It's mediated, it's supported by the complement system, but there are other cells, macrophages, platelets, NK cells, neutrophils, all those, eosinophils, they all get involved with type 2 hypersensitivity reactions as well. Both IgM and IgG are involved with type 2 hypersensitivity reactions, and specific disease conditions in which there is something wrong with a particular cell or some reason where your body detects that it needs to destroy that cell or create an immune reaction to that cell. One example of that is the ABO blood system. When somebody is exposed to a different blood type that is not compatible, your body sees that and will destroy that because it has antigen on that cell that your body does not recognize. The only exception to that is O blood. O blood doesn't have any antigens on the outside of it that your body reacts to, and so type O blood is the universal donor. Anybody can receive that. The hemolytic disease of the newborn, which you should take a look at, that is another type 2 hypersensitivity reaction. Myasthenia gravis. Myasthenia gravis is where there are antibodies produced to the receptors of at the neuromuscular junction, so that the acetylcholine can't get across from the nerves to the muscles, causing an effect, and so you have a weakness of those muscles or paralysis of those muscles. We'll talk about myasthenia gravis quite a bit. There are thyroid diseases that are caused by that are considered type 2 hypersensitivity reactions. Graft rejections, when your body does not recognize a specific cell in a tissue that's been grafted, it can create a type 2 hypersensitivity reaction where it destroys those cells, and that would be a big bummer. All right. Read more about that. Make sure, I'm just giving you the brief overall. You need to go through and study those pretty well. Type 3 hypersensitivity reactions. They're kind of similar to type 2 in that there's a reaction. In type 2, there's a reaction to a cell. Type 3 hypersensitivity reaction is where antibodies are created to antigens, and these antigen-antibody complexes are what cause the problem. Your body creates these complexes, which is really great, and then it has to clear them. And I think I've got a slide on there. Large complexes are cleared by the macrophages. They have to come along and Pac-Man phagocytize them and break them down. Small ones pass right through the kidney. Good. And the intermediate ones, those intermediate ones are a problem. They actually get deposited into tissues. And when they do, that creates a reaction in the tissue, an inflammatory reaction in the tissue, which destroys that tissue or causes damage in that tissue. Examples of type 3 hypersensitivity reactions. So this is what's happening with rheumatoid arthritis. With a lot of autoimmune diseases, they're type 3 hypersensitivity reactions. So in rheumatoid arthritis, it has less to do with the joints as it does with the deposition of these antigen-antibody complexes into the joint tissue. Let me explain what happens in rheumatoid arthritis. It's really interesting. There's a big connection to the gastrointestinal tract. There is with a lot of immune diseases. As we've talked about before, the gastrointestinal tract has a nice fine mesh, so that things that are broken down into appropriate proteins, and so that your body can do something with them, pass through these little tiny holes, like a little sieve. Well, in somebody who has an inflamed gut, who would that be? Somebody with a bad diet, somebody who's recently had antibiotics, whose gut is compromised in some way. Their gastrointestinal tract is inflamed, so they have large holes. So proteins pass through those holes without being completely digested. Your body sees them and goes, "What? We don't know what this is." Phagocytize those. Along comes antibodies that will create this antigen-antibody complex. Some of them will get cleared, most of them won't. They get deposited into joint material, causing deformed joints. So, rheumatoid arthritis is actually less about less about joints and more about the gut. So clear up the gut from the naturopathic perspective, clear up the gut, less likely to have susceptibility to rheumatoid or certainly exacerbation of rheumatoid arthritis. So SLE, systemic lupus erythematous, you need to look at that disease. It is the quintessential autoimmune condition, and those antigen-antibody complexes get deposited into a lot of tissues, and you'll need to look at what tissues those are. Drug reactions are type 3 hypersensitivity reactions. Your body, you're getting a drug, your body sees that and says, no way, well, I'm not going to deal with this drug, I don't know what to do with it, creates an antigen-antibody complex, and those get deposited into joints. Glomerulonephritis, we'll talk about glomerulonephritis. That is an inflammation of the glomerulus in the kidney. Well, when you have inflammation of the glomerulus, instead of being a nice filtering mechanism, large proteins pass through and cause damage. We'll talk about that more when we get to the renal system. However, strep infections leading to kidney trouble, glomerulonephritis, It's a type 3 hypersensitivity reaction. Your body will develop these complexes, and as I said, the small antigen antibody complexes get deposited, or I should say get cleared in the kidney. Well, yeah, if they're small, but if they're not small, they get deposited in that glomerular membrane, causing inflammation, and that destroys that filtering mechanism. But it's exposure to grains and dusts, and there's viral infections. Some diseases, which we'll talk about, something called a Henoch-Schönlein purpura, and happens when children are exposed to viruses, just a common childhood virus. These complexes will get deposited into little capillaries. They'll break open, and they'll get these broken blood vessels all over their body, called purpura, that's as a result of viral infection. Usually they're self-resolving, but they can cause major trouble. Those are all type 3 hypersensitivity reactions. Type 4 hypersensitivity reaction. Let me take a breath. Type 4 hypersensitivity does not involve antibody. It's the only one that doesn't involve antibody. It involves cytotoxic T cells, where there are substances that your body doesn't like, and you will create effector T cells, CD8s, that will cause that are stimulated by T helper 1s. Remember, will cause direct killing of cells. Poison ivy reaction, delayed hypersensitivity reactions are type 4 hypersensitivity reactions. So the inflammation from a metal of an earring causing over time an inflamed reaction, that's a type 4 hypersensitivity reaction, or a belt buckle that might be irritating the skin, or poison ivy, any kind of exposure that causes a delayed hypersensitivity reaction. Take a look at that. All right. Make sure that you understand, read the material on acute rheumatic fever. I alluded to it earlier. That's from a strep infection. Generally, it doesn't affect a lot of people, but children are at risk for the development of sequelae secondary to exposure, because what happens is there are proteins on that strep bacteria called M proteins. Your body develops antibodies to that antigen. And then what happens is it cross-reacts with your body's heart cells, because the protein on those M proteins in the bacteria, in the strep bacteria, look enough like proteins on your heart valves that your body says, when it has T cells that encounter the heart valves, the tissue in the heart valves will say, this isn't right, and it causes a hypersensitivity reaction and causes damage to those heart valves. Bummer. So make sure that you take a look at that. Look at alloimmunity. Look at Rh incompatibility. That's a type 2 hypersensitivity reaction. That's because there is another antigen, the D antigen on the cells, and they're causing a reaction. Make sure that you understand that. What happens is that the mother's immunoglobulin crosses the placenta and destroys red blood cells. So make sure that you spend some time with that one. And then again, making sure that you understand SLE, because again, it's the quintessential autoimmune disease. Autoimmune diseases affect women more commonly. There are a number of those. We'll talk about them throughout the semester, but make sure that you understand SLE. SLE is where we develop antibodies to self. We don't recognize self. That's a bummer because we don't recognize our own cells. And so we create antigen antibody complexes to our own tissue, to our own nucleic acids, to our own red blood cells, coagulated proteins, lymphocytes, platelets, those are all listed there. So when we get those, when we create an immune response, we create antibodies to our body's own antigens, our proteins on our cells. Because we have a loss of tolerance to self, we no longer recognize self. Those will create a type 3 hypersensitivity reaction, and those will get deposited in certain tissues which cause inflammation. Specifically, you'll see there in joints, about 90% in the joints, in the vascular tissue, causing rashes, renal, in the kidneys, there's hematologic changes, there's cardiovascular disease changes. I see a lot of pericarditis with those. And so make sure that you study that. Take a look at graft rejection, ABO, and then acquired immune deficiency syndrome, secondary to HIV. Make sure that you take a look at that and understand the correlation of CD4s and CD8s in HIV. Which cell is primarily infected in an HIV infection. Read about that and you'll start to understand. I think you'll be able to trace back and figure out, okay, if that is the case, if you have the loss of one of those helper T cells, what kind of diseases then are you going to be susceptible to? So HIV is simply the virus, and with that virus you get hematologic changes in the CD4-CD8 ratio, which is going to predispose you to other diseases. So people die from those related diseases. Please take a look at that. Don't memorize any of this, that slide that has a demonstration about about HIV, the virus itself, but it's really important to take a look at because you can see how they've developed drugs that have stopped certain processes, protease inhibitors, entrance inhibitors, etc. They've developed those and they've been able to control HIV because of understanding this process for how the virus gets interjected into the cells, how it gets transcribed, and then produces new copies of itself. We've been able to stop that pretty well. And then also looking at anaphylactic shock. Anaphylaxis, I think, is important. It's a type 1 hypersensitivity reaction. It's an allergic reaction and to various substances creating that IgE immune response. So I think that is the most of it. Wow, 48 minutes, pretty good. Good luck. There is no in-class lecture this week because I'm out at a conference, but make sure that you are prepared for it. Make sure that you understand the key points. Go through the slides. In the inflammation PowerPoint, I would make sure that there's probably 1 or 2 lines from each slide that's important. In the altered immunity, the one I just went through, you're going to need to understand this in a little bit more detail than what I've gone through, but hit the slides that I've particularly emphasized. Go back to my other lecture that you find in the media library, read the book, look at Khan Academy, look at the animations on YouTube. You're going to have to spend some time. The test will open on probably Thursday, I'll open it, and it'll be open until Sunday night. Remember, don't navigate from one screen to another, because that will kick you out, and it will close your test. And you really want to have every opportunity at success that you can get, because this is a tough one. Now, if you don't do well in this, don't panic yet. If you've had 2 bad tests, it's not necessarily indicative of the success you'll have. You can still do well. You can still get an A, but you really gotta be questioning what's going on with that. So that said, have a good couple of days. Congratulations at getting through this this lecture, and we'll talk with you another time soon.